Right now, somewhere deep in a laboratory, scientists are watching molecules do something that was supposed to be impossible: organize themselves, copy themselves, and begin to evolve without any living thing guiding the process. If they succeed in pushing this process just a few steps further, they will have recreated the single most consequential event in the history of the universe, the moment dead chemistry became alive. The question of how life began is not some dusty philosophical relic. It is an active, urgent, rapidly advancing field of science, and in the last few years alone, breakthroughs have shattered long-held assumptions and opened possibilities that would have seemed like science fiction a generation ago. This is the story of humanity's greatest unsolved mystery. How did lifeless matter on a violent, barren planet somehow wake up and start thinking? The ancient Greek philosopher Aristotle proposed something called spontaneous generation, the idea that living things simply spring into being from non-living matter. Flies came from rotting meat. Mice came from grain. Worms came from mud after rain. It sounds absurd today, but this was the dominant explanation in Western thought for roughly 2,000 years. No one seriously questioned it because, on the surface, it seemed to match everyday observation. Leave food out and maggots appear. Store grain in a barn and rats show up. For most of human history, that was considered sufficient evidence. The first real demolition of spontaneous generation came in 1859, when a French chemist named Louis Pasteur conducted one of the most elegant experiments in the history of science. Pasteur took nutrient broth, boiled it to kill any existing microbes, and then sealed it inside a specially designed flask with a long, curved neck shaped like a swan's throat. The curve allowed air to enter but trapped airborne dust and microorganisms in the bend, preventing them from reaching the broth. The result was decisive. The broth remained clear and sterile indefinitely. No life appeared. But when Pasteur broke the neck of the flask, allowing dust particles carrying bacteria to fall into the broth, microbial life exploded within days. The conclusion was inescapable. Life does not spontaneously arise from non-living matter under present conditions. Life comes only from pre-existing life. Pasteur's experiment settled the question for ordinary, everyday biology, but it simultaneously sharpened a deeper puzzle. If life only comes from life, then how did the very first living thing arise on a planet that, for hundreds of millions of years, had no life at all? Pasteur himself acknowledged this paradox. He had proven that spontaneous generation does not happen in a sealed flask on a laboratory bench. But he could not speak to what might have happened on a primordial Earth with radically different chemistry. An atmosphere devoid of oxygen, oceans of dissolved minerals, and energy pouring in from volcanoes, lightning, ultraviolet radiation, and asteroid impacts. That deeper question, the ultimate origin of the first living cell, remained wide open. The modern scientific assault on this question began in the 1920s, when two scientists working independently on opposite sides of the world arrived at remarkably similar ideas. In the Soviet Union, a biochemist named Alexander Oparin published a book in 1924 arguing that life had arisen through a gradual process of chemical evolution. In Britain, the geneticist J.B.S. Haldane reached almost identical conclusions in a 1929 essay. Both men proposed what became known as the primordial soup hypothesis. Their idea was straightforward in concept, even if staggeringly complex in detail. The early Earth, they argued, had a reducing atmosphere, one rich in gases like methane, ammonia, hydrogen, and water vapor, with little or no free oxygen. Energy from lightning, volcanic heat, and ultraviolet light would have driven chemical reactions among these gases, producing simple organic molecules such as amino acids, sugars, and nitrogenous bases. These molecules would have accumulated in the oceans over millions of years, creating a warm, concentrated broth, a primordial soup. Within that soup, further reactions would have built increasingly complex molecules, eventually producing self-replicating systems enclosed in primitive membranes. Haldane specifically described the ancient ocean as a hot dilute soup of organic compounds, while Oparin envisioned tiny droplets called coacervates, membrane-like structures that could concentrate chemicals and carry out rudimentary metabolic reactions. Though they used different language and emphasized different mechanisms, both men agreed on the core principle. Life was not created in a single miraculous instant, but emerged gradually through chemistry that obeyed ordinary physical laws. For nearly 3 decades, the Oppenheim hypothesis remained a compelling but untested idea. Then, in 1953, a 23-year-old graduate student at the University of Chicago named Stanley Miller decided to put it to the test. Working under the guidance of Nobel laureate Miller-Urey, Miller built an apparatus designed to simulate the conditions of the early Earth in miniature. He filled a closed system of glass flasks and tubes with a mixture of gases thought to represent the primordial atmosphere: methane, ammonia, hydrogen, and water vapor. He heated water in one flask to produce steam, simulating evaporation from the early ocean. Then he fired electrical sparks through the gas mixture, mimicking lightning. Miller let the experiment run continuously for a week, cycling the gases through the spark chamber and condensing the products back into the water reservoir. When he analyzed the contents of the water at the end of the experiment, the results electrified the scientific world. The once clear water had turned a murky reddish-brown, and chemical analysis revealed the presence of several amino acids, the building blocks of proteins and fundamental components of all known life. Miller initially identified 5 amino acids, including glycine and alanine. Decades later, when scientists reanalyzed his original sealed vials using more sensitive modern techniques, they found a total of 23 amino acids and 4 amines, including 7 sulfur-containing organic compounds, a far richer chemical harvest than anyone had initially realized. The Miller-Urey experiment was a landmark. For the first time, someone had demonstrated that the basic building blocks of life could form spontaneously from simple inorganic gases under conditions plausibly resembling those of the early Earth. It transformed the origin of life from a purely philosophical question into an experimental science. Newspapers across the United States ran headlines suggesting that scientists were on the verge of creating life in a test tube. But the excitement was tempered by limitations that became increasingly apparent over the following decades. The most significant criticism concerned the composition of the atmosphere Miller had used. His strongly reducing gas mixture, dominated by methane and ammonia, reflected the best understanding of the 1950s. But subsequent geological and geochemical research suggested that the actual early Earth atmosphere was probably much less reducing, consisting mainly of carbon dioxide, nitrogen, and water vapor, with relatively little methane or ammonia. Under these more neutral atmospheric conditions, the yield of amino acids and other organic molecules drops considerably. However, this objection turned out to be less fatal than it first appeared. Later experiments showed that even under more neutral atmospheric conditions, amino acids and other organics can still form if certain catalysts are present. Iron and nickel minerals commonly found in meteorites, for example, can catalyze the formation of organic molecules from carbon dioxide and hydrogen. Furthermore, large asteroid impacts, which were extremely common during the first several 100 million years of Earth's history, a period known as the Late Heavy Bombardment, would have temporarily generated localized pockets of highly reducing atmosphere by vaporizing iron-rich impactors and releasing enormous quantities of hydrogen. In those transient but repeated episodes, conditions very similar to Miller's flask would have existed on a planetary scale. A second limitation of the Miller-Urey experiment was more fundamental. Miller had produced amino acids, the monomers or individual building blocks, of proteins, but the leap from scattered amino acids floating in a dilute ocean to functioning proteins, let alone self-replicating cells, is enormous. Proteins are long chains of amino acids folded into precise three-dimensional shapes, and they perform virtually all the chemical work inside living cells. Getting amino acids to link together into protein-like chains, called peptides, requires removing water molecules at each bond, a reaction that is thermodynamically unfavorable in a watery environment. The British crystallographer J.D. Bernal pointed out this problem clearly, noting that producing monomers was only the first step. To explain life, you also need to explain how those monomers polymerized into functional macromolecules like proteins and nucleic acids, and how a continuous supply of energy was maintained to drive these reactions far from equilibrium. In other words, getting the ingredients was one thing, baking the cake was something else entirely. These challenges prompted scientists to explore alternative environments where life might have originated, places that could provide not only raw materials, but also concentrated conditions, sustained energy sources, and mineral catalysts to drive chemistry uphill toward greater complexity. One of the most powerful alternative hypotheses emerged from an entirely unexpected discovery on the ocean floor. In 1977, scientists aboard the deep-sea submersible Alvin, operating near the Galapagos Rift in the Pacific Ocean, discovered something no one had predicted. Thriving ecosystems of giant tube worms, clams, shrimp, and microbial mats clustered around cracks in the seafloor were superheated. Mineral-rich water gushed from the Earth's interior. These structures, called hydrothermal vents, came in two main varieties. Black smokers spew water heated to over 700 degrees Fahrenheit, blackened by dissolved metal sulfides. White smokers, or alkaline vents, emit cooler water, typically between 100 and 200 degrees Fahrenheit, rich in hydrogen and methane, through porous carbonate chimneys. The organisms living around these vents did not depend on sunlight. Instead, they ran on chemosynthesis, harvesting energy from the chemical reactions between the vent fluids and the surrounding seawater. This discovery fundamentally changed how scientists thought about the requirements for life. The deep-sea hydrothermal vent hypothesis, championed most forcefully by the geochemist Michael Russell at NASA's Jet Propulsion Laboratory, proposes that life originated not in a sun-drenched surface ocean, but in the perpetual darkness of the deep sea, specifically at alkaline hydrothermal vents. Russell's model is particularly compelling because alkaline vents solve several problems simultaneously. They provide a continuous supply of chemical energy in the form of hydrogen gas reacting with carbon dioxide dissolved in seawater, essentially the same reaction that powers many modern microorganisms. They generate steep gradients of temperature, pH, and chemical concentration across the thin mineral walls of the vent chimneys. And these gradients can drive reactions much as a battery drives current through a circuit. The porous mineral structures of the chimneys create millions of tiny interconnected chambers, natural microreactors that concentrate molecules and prevent them from diffusing away into the vast ocean. And the minerals themselves, particularly iron-nickel sulfides, act as catalysts that accelerate the very reactions needed to build organic molecules. In 2019, scientists at JPL published results of an experiment in the journal Astrobiology in which they simulated ancient ocean vent conditions by combining hydrogen-rich water, carbon dioxide-enriched seawater, and iron sulfide minerals under pressures equivalent to those found more than half a mile below the ocean surface. The experiment successfully produced formate and trace amounts of methane, both organic molecules, demonstrating that the physical and chemical conditions at ancient seafloor vents could indeed drive the formation of life's building blocks without any biological input whatsoever. The deep-sea vent hypothesis has another powerful advantage. It extends naturally beyond Earth. Jupiter's moon Europa, roughly the size of Earth's moon, is encased in a shell of ice estimated to be somewhere between 2 and 20 miles thick. Beneath that ice lies a global liquid water ocean thought to be about 50 miles deep, containing roughly twice as much water as all of Earth's oceans combined. Data from NASA's Galileo spacecraft, which orbited Jupiter from 1995 to 2003, revealed fluctuations in Europa's magnetic field consistent with a salty, electrically conductive ocean. If Europa's rocky seafloor is geologically active, and tidal heating from Jupiter's immense gravity strongly suggests it is, then hydrothermal vents very similar to those on Earth's ocean floor could be operating right now beneath Europa's ice. Saturn's moon Enceladus, a small world only about 300 miles in diameter, has provided even more direct evidence. NASA's Cassini spacecraft, during close flybys between 2005 and 2017, flew through massive plumes of water vapor and ice particles erupting from fractures near Enceladus's south pole. Analysis of these plumes revealed not only water but also sodium chloride, table salt, silica nanoparticles indicative of hydrothermal activity at temperatures exceeding 190 degrees Fahrenheit. molecular hydrogen suggesting active water rock reactions on the seafloor, and complex organic molecules with masses exceeding 200 atomic mass units. Taken together, this evidence strongly suggests that Enceladus possesses a subsurface ocean with active hydrothermal vents, and at least some of the chemical ingredients associated with life. If the deep-sea vent hypothesis is correct for Earth, then the conditions for life's origin may exist right now on at least 2 other worlds in our own solar system, and potentially on billions of similar ocean worlds throughout the galaxy. However, the hydrothermal vent hypothesis is not without its challenges. The most obvious is heat. At black smokers, water temperatures can exceed 700 degrees Fahrenheit, far too hot for complex organic molecules to survive. Most amino acids, nucleotides, and other biological building blocks break down rapidly at such extreme temperatures. This is why Russell and others have focused on alkaline vents, where temperatures are much more moderate, but even at alkaline vents, maintaining delicate molecules long enough for them to accumulate and react remains a concern. There is also the question of how the first metabolic cycles got started. Modern organisms that live on chemical energy use sophisticated protein enzymes to catalyze their metabolic reactions. But those enzymes are themselves the products of billions of years of evolution. In the very beginning, without enzymes, how did the first crude metabolic reactions sustain themselves? Some researchers have proposed that mineral surfaces, particularly iron sulfides and iron-nickel alloys, could have catalyzed primitive versions of the metabolic cycles used by modern cells. The citric acid cycle, also known as the TCA cycle or Krebs cycle, is the central energy processing pathway in nearly all living cells. Remarkably, several of the reactions in this cycle have been demonstrated to occur spontaneously on iron sulfide surfaces at elevated temperatures, without any biological catalyst. This has led to the iron-sulfur world hypothesis, proposed by the German chemist Gunnar Wachter, which envisions life beginning as a network of chemical reactions on mineral surfaces in hydrothermal environments, with metabolism emerging before genetics. While some scientists look to the deep sea for life's cradle, others turn their attention in a completely different direction, to clay. In the 1960s, a Scottish chemist named Graham Cairnsmith proposed one of the most unconventional ideas in origin of life research. He suggested that the very first self-replicating systems on Earth were not organic molecules at all, but inorganic clay crystals. Clay minerals, composed primarily of aluminum silicates, form naturally from dissolved minerals in water and grow as thin, layered crystal sheets. Cairnsmith noticed that as clay crystals grow, they can propagate structural defects, irregularities in the crystal lattice, from one layer to the next. In a sense, the pattern of defects in a clay crystal is copied as the crystal grows, much as genetic information is copied when DNA replicates. Different patterns of defects could give crystals different physical properties, for example, making them more or less prone to breaking apart and seeding new crystal growth. If certain defect patterns caused crystals to replicate more successfully than others, something resembling natural selection could operate on populations of clay crystals. In this scenario, clay crystals would have been the first organisms, not alive in any biological sense, but capable of a crude form of heredity and selection. Over time, organic molecules that happened to adsorb onto clay surfaces might have begun to participate in the replication process, eventually becoming sophisticated enough to replicate on their own without the clay scaffold. Cairnsmith called this a genetic takeover, the replacement of an inorganic replication system by an organic one. The clay hypothesis has never gained wide acceptance as a complete explanation for the origin of life, partly because it is extraordinarily difficult to test. No one has demonstrated that clay crystals undergo evolution in the Darwinian sense, and the genetic takeover mechanism remains purely hypothetical. However, one aspect of Cairnsmith's idea has been powerfully validated. Clay minerals genuinely do catalyze the formation of complex organic molecules. Experiments have shown that montmorillonite clay, a common mineral, can catalyze the polymerization of RNA nucleotides into chains up to 50 units long, far longer than what forms spontaneously in solution. Clay surfaces also concentrate amino acids and promote the formation of peptide bonds. These findings have established clay not as the originator of life, but as a plausible midwife, a mineral surface that could have helped organic molecules assemble into the complex structures needed for life to get started. Perhaps the most influential hypothesis in modern origin of life research is the RNA world hypothesis. To understand why it is so important, you need to appreciate a fundamental chicken and egg problem at the heart of modern biology. In all living cells today, genetic information is stored in DNA, and the chemical work of the cell is performed by proteins. But DNA cannot replicate without protein enzymes, and protein enzymes cannot be made without the instructions encoded in DNA. Each depends on the other. So which came first, DNA or proteins? The RNA world hypothesis cuts through this paradox by proposing that neither came first. Instead, there was an earlier stage of life in which RNA, ribonucleic acid, performed both roles simultaneously. RNA is chemically similar to DNA and can store genetic information, but unlike DNA, certain RNA molecules can also fold into complex three-dimensional shapes and catalyze chemical reactions. Just like protein enzymes, these catalytic RNA molecules are called ribozymes, and their discovery in the early 1980s by Thomas Cech at the University of Colorado and Sidney Altman at Yale University earned both scientists the 1989 Nobel Prize in Chemistry. Cech discovered that an RNA molecule in the single-celled organism Tetrahymena could splice itself, cutting out a section of its own sequence and joining the remaining pieces together, without any protein assistance. Altman found that the RNA component of an enzyme called RNase P was the catalytically active part, capable of cleaving precursor transfer RNA molecules on its own. These discoveries demolished the assumption that only proteins could be enzymes and opened the door to the possibility that RNA alone could have supported a primitive form of life. The RNA world hypothesis proposes that the earliest self-replicating systems on Earth were made entirely of RNA. RNA molecules stored genetic information in their nucleotide sequences, catalyzed the chemical reactions necessary for their own replication and survival, and underwent Darwinian evolution as copying errors introduced mutations that were subject to natural selection. Over time, DNA evolved as a more stable repository for genetic information, its double-stranded structure and the replacement of the sugar ribose with deoxyribose making it less prone to degradation, while proteins took over catalytic duties because their 20 different amino acid building blocks allow for a far greater diversity of three-dimensional shapes and chemical functions than RNA's 4 nucleotide bases. RNA was gradually demoted from star player to intermediary, relegated to its modern role as a messenger and adapter between DNA and protein. But relics of the RNA world persist in modern biology. The ribosome, the molecular machine that translates genetic information into protein in every living cell, is fundamentally an RNA enzyme. The catalytic core of the ribosome, the part that actually forges the peptide bonds linking amino acids together, is made entirely of RNA. Proteins in the ribosome play only structural and supportive roles. This is powerful evidence that at some point in the deep past, RNA was in charge. Experimental work has pushed the RNA world hypothesis from speculation towards something approaching demonstration. In 2009, Gerald Joyce and Tracy Lincoln at the Scripps Research Institute in San Diego published a landmark paper describing a pair of RNA enzymes that could replicate each other indefinitely. Each RNA enzyme catalyzed the assembly of the other from smaller RNA fragments, and the system sustained exponential growth without any protein involvement. Even more remarkably, when the researchers introduced variation into the system, providing pools of slightly different RNA fragments, the replicating RNA population evolved. Variants that replicated faster outcompete slower ones, exactly as Darwinian theory predicts. This was evolution in a test tube, driven entirely by RNA, with no DNA, no proteins, no cells, and no living organism anywhere in the system. More recent work by the same group and others has produced RNA enzymes with increasingly sophisticated capabilities, including the ability to copy RNA templates with reasonable accuracy over stretches of more than 200 nucleotides. A 2024 study described a ribozyme only 45 nucleotides long that could perform two distinct chemical reactions essential for self-replication, a striking demonstration that even very short, simple RNA molecules can possess surprisingly powerful catalytic abilities. The most significant challenge to the RNA world hypothesis is the complexity of RNA itself. An RNA nucleotide consists of three components: a sugar called ribose, a phosphate group, and one of four nitrogenous bases, adenine, guanine, cytosine, or uracil. Assembling these components from simpler precursors under prebiotic conditions has been one of the hardest problems in origin-of-life chemistry. For decades, researchers tried to build RNA nucleotides by first synthesizing each component separately and then joining them together, but the final assembly step, attaching the base to the sugar, stubbornly refused to work under plausible prebiotic conditions. This impasse was broken in 2009 by John Sutherland and colleagues at the University of Cambridge, who discovered an entirely different synthetic pathway. Instead of building ribose and bases separately and then combining them, Sutherland showed that a mixture of simple precursors, including hydrogen cyanide, hydrogen sulfide, and phosphate, could react under ultraviolet light and cycles of wetting and drying to produce activated pyrimidine ribonucleotides, the building blocks for the RNA bases cytosine and uracil, in a continuous, one-pot reaction sequence. The intermediates in this pathway are not the same as the separate components of RNA. Instead, the sugar and base are built up simultaneously, each step setting up the next. Sutherland later extended this work to show that the same starting materials and conditions could also produce amino acid precursors and lipid precursors, suggesting that all three major classes of biological molecules, nucleic acids, proteins, and membranes, might have emerged from a single unified prebiotic chemistry driven by hydrogen cyanide and ultraviolet light. This was a conceptual earthquake. It meant that the building blocks of life did not need to be produced by separate, unrelated processes and then somehow brought together. They could all flow from the same source. Even with RNA and amino acids accounted for, one critical component of life remains: the membrane. Every living cell on Earth is enclosed in a lipid membrane, a thin, flexible barrier that separates the cell's interior chemistry from the outside world. Without membranes, even a perfect self-replicating RNA molecule would be useless, because its copies would simply diffuse away into the surrounding water, and the molecular community needed for cooperative chemistry would never form. The lipid world or protocell hypothesis places membranes at the very beginning of the story. Fatty acids, simple lipid molecules consisting of a water-attracting head and a water-repelling tail, spontaneously assemble into hollow spheres called vesicles when placed in water. This is not a complex or improbable event, it is basic physics, driven by the same forces that make oil and water separate. These vesicles are essentially primitive cell membranes, and they form readily under a wide range of conditions. Experiments by Jack Szostak at Harvard University and others have shown that fatty acid vesicles can grow by incorporating additional fatty acid molecules from their environment, and they can divide when subjected to mechanical agitation, such as waves lapping on a shoreline, producing daughter vesicles. They can also encapsulate RNA and other molecules within their interiors, creating tiny compartments where chemistry can occur at much higher concentrations than in the open ocean. One of the long-standing objections to the protocell model was that fatty acid membranes are unstable in the presence of magnesium and calcium ions, which are abundant in seawater and which are also required by many RNA enzymes to function. This seemed like a fatal contradiction. The conditions RNA needs to work are the conditions that destroy the membranes it needs to be housed in. But recent research has found a surprisingly elegant solution. When amino acids or small peptides are present in solution alongside fatty acids, they associate with the membrane surface and stabilize it against disruption by divalent ions. In experiments, fatty acid vesicles that would normally fall apart in seawater-like conditions remained intact when amino acids were added. This finding is remarkable because it means that three of the most important components of life, RNA, amino acids, and lipid membranes, are not merely compatible but actively help each other. Amino acids stabilize membranes, membranes concentrate RNA, and RNA catalyzes the assembly of amino acids into peptides. There is a natural synergy among these molecules that makes the emergence of protocells seem less like an astronomically improbable accident and more like a predictable outcome of prebiotic chemistry under the right conditions. A unifying theme emerging from decades of research is that no single hypothesis, primordial soup, deep-sea vents, clay surfaces, RNA world, or lipid membranes, tells the complete story by itself. The actual origin of life was almost certainly a composite process involving elements from multiple scenarios. One plausible integrated narrative might go something like this. On the early Earth, roughly 4 billion years ago, a combination of atmospheric chemistry, volcanic outgassing, and extraterrestrial delivery from meteorites and comets provided a steady supply of simple organic molecules, including amino acids, nucleotide precursors, and fatty acids. In certain favorable environments, perhaps warm ponds subject to cycles of wetting and drying, perhaps the porous mineral chimneys of alkaline hydrothermal vents, perhaps tide pools on volcanic shores where clay minerals lined the rocks, these molecules accumulated at concentrations far higher than in the open ocean. Clay and mineral surfaces catalyzed the polymerization of nucleotides into short RNA chains and amino acids into short peptides. Fatty acids self-assembled into vesicles, trapping RNA and peptide molecules inside and creating protocells, tiny chemical reactors isolated from the diluting effect of the surrounding water. Within these protocells, RNA molecules that happen to have catalytic activity, ribozymes, gained a selective advantage because their chemical products remained concentrated inside the vesicle rather than diffusing away. Some of these ribozymes may have developed the ability to copy RNA templates, however imperfectly, setting in motion the first crude form of genetic replication. Copying errors introduced variation, and protocells containing more effective ribozymes grew faster, divided more frequently, and outcompete their neighbors. Darwinian evolution had begun. From that point forward, the trajectory, while unimaginably long and complex, follows principles that biologists understand well. Over billions of years, RNA gradually ceded its genetic storage role to the more stable DNA and its catalytic role to the more versatile proteins, retaining its position as the essential intermediary between the two. Single-celled organisms diversified, developed photosynthesis, oxygenated the atmosphere, and eventually gave rise to the staggering profusion of multicellular life that inhabits the planet today. The question of where all this happened, which specific environment on the early Earth served as life's cradle, remains hotly debated. The traditional image of Darwin's warm little pond has been updated and expanded considerably. Recent experiments have drawn attention to the importance of wet-dry cycling, the repeated wetting and drying of surfaces, as would occur on shorelines, in hot springs, or in evaporating pools. A 2024 study published in the Proceedings of the National Academy of Sciences demonstrated that wet-dry cycles can cause nucleic acid monomers to polymerize into chains without the chemical activation agents that previous experiments had required. The repeated drying concentrates the molecules and removes water, driving bond formation, while the re-wetting disperses the products and allows them to interact in new combinations. When researchers at the Santa Fe Institute and collaborating institutions mimicked these cycles in laboratory settings designed to replicate early Earth conditions, they observed something unexpected. Molecules did not react randomly. Instead, they self-organized into predictable patterns of increasing complexity. Chemical evolution under periodic environmental forcing was not the chaotic, purely random process that many had assumed. It was orderly, directional, and, at least to some degree, predictable. This finding challenges the long-held view that life's origin was a fantastically improbable accident. If prebiotic chemistry naturally tends towards self-organization under the right environmental rhythms, then the emergence of life may be less like winning the cosmic lottery and more like water flowing downhill, a natural consequence of physical and chemical laws operating under specific conditions. The contribution of extraterrestrial material to Earth's prebiotic chemistry has become increasingly well documented. The most dramatic recent evidence comes from NASA's OSIRIS-Rx mission, which collected samples from the surface of the asteroid Bennu, a carbon-rich near-Earth asteroid about 1/3 of a mile in diameter, and returned them to Earth in September 2023. Analysis of the Bennu samples, published in the journals Nature and Nature Astronomy, revealed an astonishing chemical inventory. Scientists detected 14 of the 20 amino acids that life on Earth uses to build proteins, plus all 5 nucleobases that DNA and RNA use to encode genetic information: adenine, guanine, cytosine, thymine, and uracil. They also found exceptionally high concentrations of ammonia, formaldehyde, which can react with ammonia to form amino acids, and traces of 11 different evaporite minerals, salts left behind by the slow evaporation of liquid water, indicating that Bennu's parent body once hosted a long-lived hydrothermal system with circulating brines. Danny Glavin, a senior sample scientist at NASA's Goddard Space Flight Center and co-lead author of the Nature Astronomy paper, emphasized that these findings would not be possible without a sample return mission, meticulous contamination control measures, and careful curation and storage of this precious material. The significance of the Bennu results extends far beyond a single asteroid. If a small, seemingly barren space rock orbiting between Earth and Mars contains this rich a cocktail of life's building blocks, then such molecules are likely common throughout the solar system, and, by extension, throughout the galaxy. As Glavin noted, the fact that life's building blocks are manufactured in space and distributed widely increases the odds life could have formed on other planets and moons. The idea that life itself, not just its chemical ingredients, might travel between worlds is the essence of the panspermia hypothesis. The word comes from Greek, meaning seeds everywhere, and the concept has a long intellectual history, dating back at least to the Greek philosopher Anaxagoras in the 5th century BCE. In its modern form, panspermia proposes that microorganisms or their hardy spores could survive the journey through space inside meteorites, comets, or interplanetary dust particles, seeding new worlds with life. This is not as far-fetched as it might sound. Experiments conducted on the International Space Station have shown that the bacterium Deinococcus radiodurans, nicknamed Conan the bacterium for its extraordinary toughness, can survive at least 1 year of direct exposure to the vacuum, extreme temperature swings, and intense cosmic radiation of low Earth orbit. On Earth, this organism can withstand radiation doses thousands of times greater than what would kill a human, because it possesses remarkably efficient DNA repair mechanisms that can reassemble its shattered genome within hours of exposure. Bacterial spores of certain bacillus species are even hardier, capable of surviving in a dormant state for millions of years. The late heavy bombardment, which pummeled the inner solar system with asteroid and comet impacts roughly 3.8 to 4.1 billion years ago, would have launched enormous quantities of rock from one planet's surface to another. Calculations show that material blasted off the surface of Mars by a large impact can reach Earth in as little as a few months, and some fraction of that material would experience only moderate heating during the transit, potentially survivable for microorganisms embedded deep within the rock. The famous Martian meteorite ALH 84001, recovered from Antarctica in 1984 and found to have originated on Mars based on the isotopic composition of gases trapped in its interior, ignited worldwide excitement in 1996 when a team of NASA scientists announced that it contained structures resembling fossilized microorganisms and mineral features consistent with biological activity. Subsequent analysis strongly suggested that these features were more likely the result of inorganic geological processes. But the controversy highlighted how thin the line between life and non-life can be at the microscopic scale, and how desperately humanity wants to find evidence that we are not alone. Panspermia, it should be noted, does not actually solve the origin of life problem, it merely relocates it. If life on Earth came from Mars, the question becomes, how did life originate on Mars? If life arrived on a comet from another star system, we need to explain how it started there. At some point, somewhere, life had to emerge from non-living chemistry through the processes described in the preceding sections. Panspermia does, however, have profound implications for how we think about the distribution of life in the universe. If life can travel between planets and even between star systems, carried by meteorites ejected by impacts, or perhaps aboard interstellar objects like Oumuamua, the mysterious cigar-shaped body that passed through our solar system in 2017, then a single origin event on one world could seed an entire galaxy over billions of years. The question would shift from how often does life originate to how far can it spread. The effort to create life from scratch in the laboratory, synthetic biology, represents the flip side of origin of life research. Instead of looking backward to deduce how life began, synthetic biologists look forward, attempting to build living systems from non-living components and, in the process, testing which ingredients and conditions are truly essential. The most famous milestone in this field came in 2010, when a team led by the American geneticist Craig Venter at the J. Craig Venter Institute announced the creation of the first synthetic cell. Venter's team chemically synthesized the complete genome of a bacterium called Mycoplasma mycoides, about 1.08 million base pairs of DNA, entirely from laboratory chemicals, one nucleotide at a time. They then transplanted this synthetic genome into the emptied cell of a closely related species, Mycoplasma capriculum, whose own DNA had been removed. The recipient cell, now running on an entirely human-designed genome booted up, began expressing the synthetic genes and started dividing. The synthetic cells were indistinguishable from natural Mycoplasma mycoids in their behavior. Venter described the achievement as the first self-replicating species we've had on the planet, whose parent is a computer. The work provoked intense debate. Some hailed it as a breakthrough that would lead to designer organisms for medicine, energy, and environmental cleanup, while critics warned of the dangers of playing God and the potential for engineered organisms to cause ecological havoc. In 2016, Venter's team went further, creating JCVI, SYN 3.0, a minimal cell containing only 473 genes and 560 base pairs of DNA, the smallest genome of any self-replicating organism known. This stripped-down cell can grow and divide, but it contains no unnecessary genes. Every one of its 473 genes either has a known essential function, or is one of 149 genes whose function remains mysterious, but whose removal kills the cell. The existence of JCVI, SYN 3.0, tells us something important about the minimal requirements for life. Even the simplest conceivable free-living cell needs at least several hundred genes, encoding thousands of coordinated molecular interactions. This underscores just how enormous the gulf is between prebiotic chemistry and even the most primitive living cell, and how much remains to be discovered about the steps that bridge that gulf. The philosophical question lurking behind all of this science is deceptively simple. What is life? It is a question that has occupied thinkers from Aristotle to Schrödinger to the designers of Mars rovers, and it still has no universally accepted answer. The most widely cited working definition was proposed by a NASA committee in 1994. Life is a self-sustaining chemical system capable of Darwinian evolution. This definition has the virtue of being concise and capturing two of life's most distinctive features: metabolism, self-sustaining chemistry, and evolution, heritable variation subject to natural selection. But it also has awkward edge cases. Viruses evolve through natural selection and carry genetic information, but cannot reproduce on their own. They require a host cell's machinery. Are they alive? Most biologists say no, or at least not quite, but the answer is far from settled. Mules are alive but cannot reproduce. Does that disqualify them? Fire is self-sustaining and spreads, but it does not evolve. Computer viruses replicate and even mutate, but few would call them alive. A crystal grows by adding material in a pattern dictated by its structure, a process superficially resembling reproduction, but it lacks metabolism and evolution. Every proposed definition of life has exceptions and borderline cases, and the difficulty of drawing a bright line between living and non-living may itself be telling us something important. That the transition from chemistry to biology was not a sharp boundary crossing, but a gradual continuum. A spectrum along which increasingly complex chemical systems acquired increasingly lifelike properties until, at some point, they crossed an invisible threshold that we retroactively label alive. The physicist Erwin Schrödinger, in his extraordinarily influential 1944 book, What is Life?, approached the question from the perspective of thermodynamics. He pointed out that living organisms appear to violate the second law of thermodynamics, the law that says entropy, or disorder, always increases in a closed system. Living things maintain exquisite internal order, precisely folded proteins, intricately organized genomes, meticulously regulated metabolic pathways. How can this be, in a universe that relentlessly tends toward chaos? Schrödinger's answer was that organisms are not closed systems. They maintain their internal order by importing low-entropy matter and energy, food, sunlight, from their environment and exporting high-entropy waste, heat, carbon dioxide, excrement back to it. They do not violate the second law, they obey it, at the expense of increasing entropy in their surroundings. In Schrödinger's memorable phrase, organisms feed on negative entropy. This thermodynamic perspective reframes the origin of life as a question about how self-sustaining, entropy-exporting chemical systems first arose. Norbert Wiener, the MIT mathematician who founded the field of cybernetics, the study of control and communication in animals and machines, took the information-theoretic view even further. He argued that the essential feature of both living organisms and well-designed machines is their ability to gather information from the environment, process it, and use it to maintain their own organization through feedback loops. Wiener saw no fundamental difference between a thermostat regulating a building's temperature and a bacterium sensing and swimming toward a food source. Both are information processing systems using feedback to maintain a desired state. This perspective, radical in the 1940s and 1950s, has become increasingly mainstream. Modern biology routinely describes life in informational terms. DNA stores information. Ribosomes decode it. Cells process environmental signals through molecular signaling networks. And brains perform massively parallel computation using electrochemical signals. If life is fundamentally about information processing, then the question of whether a sufficiently sophisticated artificial intelligence could be considered alive is not merely science fiction. It is a genuine philosophical frontier. The search for life beyond Earth is no longer a theoretical exercise. Multiple missions are actively looking, or are being designed to look, for biosignatures, chemical or physical signs of life, on other worlds. NASA's Perseverance rover, which landed on Mars in February 2021, has been collecting rock core samples from the ancient lakebed of Jezero Crater, a site chosen specifically because it once held a body of liquid water and may preserve signs of ancient microbial life. These samples are intended to be returned to Earth by a future mission for detailed laboratory analysis, the most sensitive search for Martian life ever attempted. The European Space Agency's ExoMars program aims to drill up to 6 feet into the Martian subsurface, where organic molecules would be shielded from the intense ultraviolet radiation that sterilizes the surface. NASA's Europa Clipper mission, launched in October 2024, is heading to Jupiter's moon Europa, where it will perform nearly 50 close flybys, mapping the ice shell, measuring its thickness, detecting possible plumes of water erupting through cracks, and searching for the chemical signatures of a habitable ocean beneath. The Dragonfly mission, scheduled to launch in the coming years, will send a nuclear-powered drone to the surface of Saturn's moon Titan, where it will fly from site to site, analyzing the exotic organic chemistry of a world with methane rain, ethane lakes, and a thick nitrogen atmosphere, a world that may preserve a snapshot of the kind of prebiotic chemistry that preceded life on Earth, frozen in time by Titan's extreme cold. Meanwhile, the James Webb Space Telescope and planned next-generation observatories are beginning to characterize the atmospheres of exoplanets, planets orbiting other stars, looking for combinations of gases that would be difficult to explain without biology. Oxygen and methane, for example, are chemically reactive and would destroy each other within geological time unless they were being continuously replenished, as they are on Earth by photosynthesis and methanogenesis, respectively. Detecting both gases simultaneously in the atmosphere of a rocky planet in the habitable zone of its star would be strong, though not conclusive, evidence of a living biosphere. The sheer number of potential targets makes the search encouraging. Studies based on data from NASA's Kepler Space Telescope, which surveyed a small patch of the Milky Way from 2009 to 2018, estimated that the galaxy contains between 11 billion and 40 billion rocky planets in the habitable zones of their stars, the region where liquid water could exist on a planet's surface. Even if the probability of life arising on any given world is extremely small, the number of rolls of the dice is staggeringly large. The famous Drake equation, formulated in 1961 by the American astronomer Frank Drake, attempts to estimate the number of communicative civilizations in the galaxy by multiplying together a series of factors: the rate of star formation, the fraction of stars with planets, the number of habitable planets per star, the fraction on which life actually arises, the fraction on which life develops intelligence, the fraction that develop technology capable of interstellar communication, and the average lifetime of such civilizations. The equation is more a framework for organized thinking than a precise calculation. Most of its factors are unknown or poorly constrained, but it highlights a crucial point. If the fraction of habitable planets on which life arises is close to 1, that is, if life is an almost inevitable consequence of the right chemistry in the right environment, then the galaxy should be teeming with biology. The Fermi paradox, named after the Italian-American physicist Enrico Fermi, poses the uncomfortable counterpoint: if life and intelligence are common, why have we not detected any evidence of extraterrestrial civilizations? Proposed resolutions range from the plausible: intelligent life is common, but interstellar communication or travel is prohibitively difficult, to the sobering: most civilizations destroy themselves through war, environmental collapse, or technological catastrophe before they can make contact, to the exotic: we are living in a simulation, or advanced civilizations deliberately avoid detection. The absence of evidence is not evidence of absence, but it is a data point that any comprehensive theory of life in the universe must eventually address. As humanity stands on the threshold of potentially creating life from non-living matter in the laboratory, detecting life on another world, or building artificial intelligences that blur the line between living and non-living, the ethical dimensions of this quest become increasingly urgent. The creation of synthetic organisms raises questions about safety. What happens if an engineered microbe escapes the lab and competes with natural organisms in the environment? And about responsibility, do we have a moral obligation to the organisms we create, particularly as they become more complex? The detection of extraterrestrial life, even microbial life, would be among the most consequential discoveries in human history, fundamentally altering our understanding of our place in the cosmos and potentially raising questions about planetary protection, our obligation not to contaminate pristine extraterrestrial ecosystems with hitchhiking Earth microbes, or to endanger Earth's biosphere with alien organisms brought back by sample return missions, and the development of artificial intelligence systems capable of self-improvement and open-ended. Learning raises the question of whether we might one day need to extend our definition of life and our ethical frameworks to include entities made of silicon and code rather than carbon and water. The story of life's origin is, at its deepest level, the story of information. In the beginning there were atoms, hydrogen, helium, lithium, forged in the furnace of the Big Bang roughly 13.8 billion years ago. Those atoms coalesced into stars, and within those stars, nuclear fusion built heavier elements: carbon, nitrogen, oxygen, phosphorus, sulfur, the elements of life. When massive stars exploded as supernovae, they scattered these elements across the galaxy, seeding molecular clouds that collapsed to form new stars and planets. On at least one of those planets, a small, rocky world orbiting an unremarkable yellow star in an outer arm of the Milky Way, those elements combined into molecules of increasing complexity. At some point, roughly 4 billion years ago, one of those molecular systems crossed a threshold. It began to copy itself. It began to make errors in copying, producing variants. Some variants were better at copying than others, and they proliferated. Information, the sequence of nucleotides in a strand of RNA. the arrangement of amino acids in a peptide, the composition of a protocell membrane began to matter. It began to be preserved, transmitted, and refined by the relentless logic of natural selection. That was the moment chemistry became biology. That was the moment the universe began to know itself. About 4 billion years have passed since that moment, and the descendants of that first self-replicating molecular system have spread to every habitable niche on this planet, from the crushing darkness of the Mariana Trench to the frozen valleys of Antarctica, from boiling acid springs in Yellowstone to the upper atmosphere, from deep rock fractures miles below the surface to the canopies of tropical rainforests. A single unbroken thread of molecular heredity connects every organism alive today, every bacterium, every fungus, every plant, every animal, every human being, back to that first primordial replicator. The genetic code is essentially universal, with minor variations. Every living thing on Earth uses the same 20 amino acids, the same 4 DNA nucleotides, the same fundamental machinery of transcription and translation that was established in the earliest cells. We're all cousins, separated by varying degrees of deep time, but united by common chemistry and common ancestry. The oldest known fossils, stromatolites, layered mounds of sediment built by photosynthetic microbial communities, date to approximately 3.48 billion years ago, found in the Dresser Formation in Western Australia. Chemical signatures consistent with biological carbon processing have been detected in rocks from Greenland dating to about 3.7 billion years ago. Given that the Earth itself is only about 4.5 billion years old, and was subjected to sterilizing asteroid bombardment until roughly 3.8 to 4.0 billion years ago. The window between when life became possible and when it apparently appeared is strikingly narrow, perhaps only a few 100 million years, and possibly much less. This has led many scientists to conclude that the origin of life, far from being a vanishingly improbable fluke, may have been a relatively rapid and perhaps even inevitable process once the conditions were right. The coming decades promise to be the most exciting in the history of origin of life research. Laboratory experiments are closing in on the creation of self-replicating chemical systems from simple precursors. Missions too much, Europa. And Enceladus may detect biosignatures or even living organisms on other worlds. Telescopes are beginning to sniff the atmospheres of distant exoplanets for signs of biological activity. Artificial intelligence is being deployed to search vast chemical parameter spaces for self-organizing reactions that might mimic prebiotic chemistry. Synthetic biology is constructing ever simpler artificial cells, working backward from modern life toward the minimal requirements for a self-sustaining, evolving chemical system. Somewhere at the intersection of all these lines of inquiry, the answer to humanity's oldest question may finally come into focus. How did life begin? We do not yet know for certain. But we are closer to knowing than at any point in the 4 billion year history of life itself. And the answer, when it comes, will reshape not only science but our understanding of who we are, where we came from, and whether we are alone in this vast and ancient universe.